My name is Katheleen Gardiner, and I'm a professor in the Department of Pediatrics at the University of Colorado in Denver, and I'm also a member of the Intellectual and Developmental Disabilities Research Center. And the major focus of my research has been and is Down syndrome.
Wonderful. And you have a very interesting approach, I know, to studying the pathways of the effect of the trisomy on the entire genome and not just the trisomy, not just the 21st chromosome?
Mm-hmm. Yes. So the logic behind what we do stems from looking at the complexity that results from an extra copy of a whole chromosome. So we know from our work, we look at the DNA on the chromosome, there are over 500 genes on chromosome 21. If all of these, or even a large number of these, are overexpressed or used more by the cells because there are 3 copies of them rather than 2 copies, then that's a lot of abnormalities, a lot of perturbations. And so one way of looking at trying to conquer the effects of trisomy 21 is try to target the genes on chromosome 21 and control them, return them to normal. But if you think about it logically, if you're dealing with 100 or 200 or maybe more genes that need to be turned down, what are you talking about, 100 or 200 drugs? No. So what you do is you ask, what do these genes on chromosome 21 do? How do they work together? And we know in many cases they do work together. So what we do is we look a little bit on 21, but most of our work is looking at what's happening to proteins that come from other chromosomes, other genes on other chromosomes. And so we are trying to integrate the effects of the overexpression or the overuse of many, many genes on chromosome 21. And then you can go downstream, and the idea is that with a single drug, you can correct the effects of overexpression of many genes. So that's why we look at pathways. We ask, what pathways do the genes on chromosome 21 affect? And therefore they affect them more in Down syndrome because they're overexpressed. We ask if those pathways are perturbed, and then once we know that, we ask, are there drugs that would let us turn the pathway down if it's too high, or turn it up if it's too low?
So how close are we, do you think, to some significant breakthrough, or what do you believe is the most essential factor to present to be able to move to the next level in research toward actual treatment and cure?
So I think in the last 3 to 4 years, there have been major advancements in a lot of areas that can be incorporated into Down syndrome research. So we know more about the pathways. We know more about drugs that affect those pathways. We know more about abnormalities that are seen in some mouse models of Down syndrome. So there are a lot of things. We know more about the human genome. We know more about the genes on chromosome 21. So now and that gives us great promise. We know that we can manipulate in a mouse model of Down syndrome some of the learning and memory deficits that the mouse displays. And those deficits are relevant to what we see in children and adults with Down syndrome. So we know we can manipulate these deficits in mouse. Okay, the problem is the mouse is not a good model because it's not a complete trisomy. It's only about 50%. So we need more models. And then it's a matter of pushing forward. How many drugs do you want to look at? I would suggest that you don't want to look at 3 or 4 or 5 and say, oh, you know, these 2 worked. I would say you want to look at tens to hundreds, maybe thousands, and then pick the best ones. So we need to think carefully about the model systems we want to use. We need to incorporate some type of human cell system. We can't, obviously, don't experiment on people, but we can take now cells, skin cells, blood cells from people, and create model systems that we can study. We can ask, what are the effects of these drugs on what we know of the pathways in these cell systems, and then maybe move into mice, and then maybe move into clinical trials. So what Down syndrome research now is poised to do is to make big advancements that will have, I think, very positive consequences for correcting at least some of the learning and memory challenges in people with Down syndrome. But it needs a focus. You know, it needs a focused, larger scale effort than the community has been able to muster so far. There's a lot of things that could go into this now to make it a big success in the next 3 to 5 to 10 years.
And you're one of the speakers here at the International Conference. What is your association? How long have you been associated with the Lejeune Foundation, and in what ways?
The Lejeune Foundation has been extremely supportive of research in Down syndrome and in other causes of intellectual disability, but they have been a constant supporter of my work. I often think of them as the people who care when nobody else seems to, because they've consistently supported projects in my lab. Pilot projects where you need to generate a little bit of information so you can go to a funding agency like the National Institutes of Health. They funded some of my postdoctoral fellows, people with a brand new PhD who are trying to get started. They really encourage Down syndrome research and have done this in a very consistent way. And they're very broadly thinking in the types of projects that they will focus on.